Good morning, all. Uh, uh, Welcome to today's talk, that is the online modern mycological lecture series titled Genome Analysis of Fungal Pathogens of Cereal Crops by Dr. Sudhir Nawathe. So the talk is, as you, those who have joined for the first time, so the talk is being a joint activity of the CBEC, that is Center for Biodiversity Exploration and Conservation, uh, and Mycoasia. So Mycoasia is a journal that is started in 2019, which is a peer-reviewed online journal, which is focusing on quality research on fungal diversity, ecology, phylogeny, and taxonomy. CBEC is conducting research on fungal fungal biology as well as taxonomy ecology. Apart from that, I would like to brief you about Myco India, which is a recent uh, kind of endeavor. So, where in the Myco India is having several activities, including the service of fungi ID also. That's a very good service. People are taking benefit of it, wherein people share their photographs of mushroom or microscopic fungi and try to get a basic idea about the fungi. Apart from that, it is also publishing some good articles and for the benefit of the students and the general public and young researchers. So far it is published online 3 articles. They are very good and important for the young researchers and students. For today's talk, I'll request all to mute yourself during the talk. Questions will be taken at the end, and or you can type your questions in the chat box or raise your hand at the end of the talk uh, when it is allowed or permitted. To brief you about today's speaker, Dr. Sudhir Nawathe. He is working as scientist C at Genetics and Plant Breeding Division at Agarkar Research Institute, Pune. He has obtained his PhD from Banaras Hindu University in mycology and plant pathology. His academic pursuits encompasses the fields of plant pathology, fungal biology, host pathogen interactions, and disease management. He also demonstrates a keen interest in the field of fungal biology, especially focusing on the topics such as fungicide resistance and related concerns to hemibiotropic and biotropic fungal plant diseases. He is presently engaged in the research pertaining to investigating the dynamics of host pathogen interaction and disease resistance mechanisms, as well as he is also engaged in the selection of the breeding of wheat, moong beans, and soybean crops with a focus on enhancing resistance to both biotic and abiotic stress factors. So far, he has published more than 40 research papers, and he's also been visiting scientist at Genetics and Plant Breeding International Maize and Wheat Improvement Center, Nairobi, Kenya, in 2022. He's also an editor of Myco India, that on the pathogenic fungi division. I welcome you, Dr. Sudhir, for the talk, and I also welcome all the participants who are attending today's talk. Thank you so much. Dr. Sudhir, you can please go ahead with your presentation. Oh, I have to share. I'll be sharing it. Yeah.
Yeah. So while Rohit sir shares the presentation, first of all, thank you for the opportunity, CBEC and Mycoasia and Dr. Shana sir, for giving me opportunity to interact with dynamic forum of scientists, very senior scientists and young researchers also. So I'll be talking on genome analysis of fungal pathogens, mostly on cereal crop. Sir, next please. So before we go ahead with slide, we'll have brief introductory parts about genome analysis. So there are several genome resources available online, and those gives us a brief idea about and insights about the various fungal pathogens and genomes in species list. They are enlisted here. So most genome popular genome resource that has that is rich in genome or various diverse genome is genome online database, what we call is gold. And there is another website, ZGI genome portal, that is also called microcosm. And then NCBI, which has more than 3,000 genome in gold, and more than 1,000 genomes in microcosm and NCBI genome. Next, please, sir. So, when we talk about the sequenced fungal genomes, there are more than 1,000 fungal genomes. Those are sequenced, and most of them are from Basidiomycota and Ascomycota. So this information is based on this genome resources available on the various database. When I say 1,090 genome, those include 1,090 species fungal genus are sequenced yet. Next, please, sir. So when we compare genomes, those are sequenced in Basidiomycetes. Most genome sequence are from Agaricomycotina, and in Ascomycetes, most of the genomes are from Pezizomycotina. Next, please, sir. So when there are genome sequence, most of sequence genome in fungal genomes are at least 50% genome sequenced are of plant pathogen, then followed by opportunistic pathogens, then parasites, and then followed by medically important and animal pathogens. And when we see their category wise, so most predominant genomes are of are decomposers, then pathogens of plants, and medically important fungal pathogens, and then some are symbiotic and Niche specific. So Niche specific refer to the species of the fungus. Those are occupying the abiotic niches, whereas symbiotic species are associated with other organism. And economically important species have use in economy, like biocontrol agents, or in pharmaceutical industries, or in culinary purpose. Next, please, sir. Uh, when genomes of fungal plant pathogens categories, there are more than 48 fungal genomes available. Those are infectious on cereal. And then if we talk about food crop pathogen, then the more than 117 genomes are sequenced in food crop pathogen. Out of that, 48 are of cereal pathogens, and remaining are pathogens of fruits, vegetables, legumes, and some are perennial crops like citrus and many more. So the genomes majority focused sequencing is happened in case of cereal pathogens. Next, please, sir. When we talk about genome size and number of operidine frame in comparison with all other genomes and plant pathogens, then most of the plant pathogens genome size is around average genome size is around 48 to 50 MB. um, compared to other, and some have less than 50 MB. And ORFs are more on an average 250 ORFs are has been presented or reported by several genomes in both ascomycete and basidiomycetes compared to all fungal genomes available in the database. Next, sir. Next, please. So some of the major fungal pathogens, those are infectious on cereal and whose genomes are most studied includes Plumeria graminis and Zymoseptoria tritici, then Pyricularia graminis tritici, then Magnaporthe oryzae, then Fusarium graminearum, then Bipolaris sorokiniana, against. Sir, next please. So when we see insides of the genome, there are some pathogens like Bipolaris sophiana that is infectious on wheat and barley, and lifestyle is hemibitroph, and average size of genome is around 37 MB. Uh, then most devastating pathogen or threatening pathogen Vaccinia graminis tritici on wheat and of concern of food security has around genome size of 89 MB. Then Zymoseptoria tritici, the most studied genome of pathogen of wheat has around and is a hemibitrophic pathogen has a genome size of around 40 MB and has several accessory chromosomes and is well known pathogen. Next slide, please, sir. So there is another most studied genome and is infectious across the world and is well studied is Plumeria graminis tritici is also a pathogen of host. It has a biotrophic life cycle and is a most largest genome of among the cereal pathogens is a Plumeria graminis and is has a genome size of around 180 MB. And then another major pathogen is Magnaporthe oryzae, and Fusarium graminearum has a genome size around 41 to 36 MB. Next, please, sir. So when we go for genome sequencing, what are the possibilities or tools available for the genome sequencing? Uh, So when we go for within the species comparison of genome sequencing, those include the steps of whole genome sequencing or genotyping by sequencing to know their mode of reproduction, demography, selections within the species, or gene flow means a gene related to pathogenicity and those have evolution or what we say has a divergent time of a particular pathogen that can be studied using the genome analysis within the species. And if we compare the genome between the species, then we would have to do the de novo assemblies and genome annotation, and that will help us to know about analysis of ortholog, then selection of the species across the continents, then any transposable element related to pathogenicity or not related to pathogenicity, then selection of specific species across the regions worldwide, and then genome size or expansion of genome size, and then phylogenetic reconstructions we can do. So when we go for analysis or comparison between two species genome, the most question arises or the question those can be answered includes what are the genetic basis of host specialization. For example, if we take example of Puccinia graminis, it has a host specialization on wheat as well as on barley and Cereal cerealis. So Psi and Puccinia graminis hordi is the two species. Those has a specialization on barley and wheat. So what is the genomic or genetic basis of this host specialization? If we talk about coexisting pathogen, is there any hybridization and emergence of new species or new races of the pathogen? That questions can be answered. Or there is, if any example or evidence of horizontal gene transfer, that also can be studied. So horizontal gene transfer has a very good example. I will explain in upcoming slide how this question was answered. Then, if we talk about, or if we and compare or do the analysis of genome within the species, so we can have answer of several questions like what is the standing genetic variation within the species, or what is the genome organization affect the evolution of virulence of that particular species or races of a particular species. Then what is the population structure within the region, and what are the factors imprints those are affecting the existing population structure, or a selection of that pathogen, or adoption of the pathogen, or the what are the environmental association that is affecting the life cycle of that particular pathogen. Next, please, sir. Then major comparative genomic analysis use are for a comparison of genome content. That includes analysis of genome size, and then whether the contents of the genome that is related to pathogenic or non-pathogenic lineage. For example, the genome expansion due to transposable elements or differences in the genome comparison or lineage specific region can be analyzed using the comparison of two genome, that is pathogenic versus non-pathogenic. Then if we compare the or do the broad comparison of the genome, those has a diverse life cycle, like biotrophic compared with hemibiotrophic, or biotrophic compared with against necrotrophic. So this will may reflect a number of genes. Those has specific functional categories. For example, effectors or cell wall degrading enzymes. Those are necessary for pathogenicity in specialization. Host specialization includes evolution of the host. That evolution of host specialization are driven by the changes in the effector genes. These host specialization by the pathogen just because of their changes in effector molecules. Those are secreted by the pathogens uh, during the infection, and these can be compared using genomes of different life cycle, or within the species host specialization. Just example is Puccinia graminis tritisae and Puccinia graminis horda. We can have know the changes in effector molecule, why this host specialization is occurring in this particular pathogen. Then host jump. So the host is including is associated with a horizontal transfer of a particular pathogenicity related gene. One a typical example of this uh, host jump is tox A gene. I will come again with to tox A gene in next slide. That is a typical example where this gene tox A gene has transferred from one species in two different species, two different genus. So this is arise due to hybridization of two pathogen or coexistence of two pathogen on the same host. So this kind of analysis can be, or this kind of information can be obtained in comparative genomics analysis in uh, using pathogen lineage. So when we talk about, sir, next please, sir. When we talk about fungal pathogen genome, their size varies from 20 MB to 180 MB. MB just we have seen the size of genomes of various content from 10% to 90% of each genome, and they may vary in chromosome numbers, gene numbers from 5,000 to 20,000, and may have compartmental compartments like accessory chromosomes and accessory regions. Next, please, sir. So when we do a genome sequencing and analyze the genome, there are several characteristics of genome compartments. For example, in core genome, uh, that can be compartmentalized based on gene density. So, if we take an example of Leptospirea maculans or a Zymosepteria tritisae, the gene regions are sparse in Leptospirea maculans, but that is mm, close or dense in case of Zymosepteria tritisae. Then transposable element contents. So Zymoseptoria tritisae has high transposable elements compared to any other genomes, and has lineage specific transposable element. Then signs of correlated evolution or accumulation of virulence related genes. If we talk about genome of smut fungi, it is highly evolving genome and has several rapidly evolving genes in the smut fungi. And then epigenetic markers. That is also a one typical characteristics, but that is confined to Zymoseptoria tritisae only, where heterochromatin enrichment on accessory chromosome is well known and high. Next. So, what is the workflow of the genome sequencing? So first step is we must have the pure culture, and then genomic DNA extraction, followed by a small or a large fragment library. This library is majorly based on the what kind of platform we are obtaining, and what is our target genome, and what is the target, or what is the objective, whether we are resequencing, we are doing a de novo assembly, completely new genome, or we want to compare only a certain part of genome. Then we must have to choose the platform for the genome sequencing. So most famous platforms are Illumina HiSeq, then Nanopore, MiniIon, and PacBio platform. And once we have the sequence, then we have to go for quality control. And quality control includes the trimming of the genome based on least informative sites, and other things. And then followed by genome assembly, then functional annotation of the genome, then genomic structure studies, pathogenic microbiology, and comparative genome analysis, what I detailed in earlier slides. Next, please, sir. So, what are the requirement of the basic genomic DNA? That is, DNA purity on OD is 1.8 to 2 without degradation or RNA contamination. If you are going for Illumina or Nanopore platform, the amount of DNA should be more than 2 microgram and concentration of around 20 nanogram per microliter. And if we are obtaining the much advanced platform like PacBio, then DNA amount what we want to have is like more than 10 microgram, and concentration needs to be very high, like more than 80 nanogram per microliter. Next, sir. So this is the most common workflow that includes the genome sequencing. Figure includes the genome sequencing up to raw data, and B includes the once we have the raw data, what we have to do? We have to do the filter and quality control. Then open taxonomic unit demarcation and taxonomic identification of genome. Then diversity of that genome. We can compare the genome using several uh, platforms, or several indicators, including Shannon diversity includes, or K1 estimator, like that. Then composition analysis in taxon summary, and statistics of the genome, followed by comparison of the genome to visualize, and then some clustering analysis and all. Next, please, sir. So, what is the when we do the analysis? What are the basic information is to be presented? First is a sequencing platform. Then number of read we obtained out of the genome. They may be in millions, or they may be in thousands. Then number of reads of the sequencing. Then total length of the genome. Then total read sequence and coverage. And when we talk about assembly statistics, total genome size in base pair, then number of contigs, number of scaffold, then GC content, N50. N50 is length of smallest scaffold in order set of scaffold corresponding to 50% of total genome size. And then another criteria is L50, that is the smallest number of scaffold whose length equals to 50% of total genome size. Then BUSCO is another criteria that indicates the completeness of genome. And then annotation, that is number of genes, secondary metabolites, and key enzymes, then carbohydrate active enzymes, then number of secreted proteins or number of predicted proteins, and number of candidate effectors molecule available in that particular genome. Next, please, sir. So, when we talk about genomic assembly, we can analyze the uh, what are the coding and non-coding gene and predict them. Then we can also analyze the repetitive sequence analysis. Then in genetic genomic structure analysis, that includes genomic isolate production prediction, then detection of CRISPR, linked RNA prediction, then in functional annotation, gene ontology, then several database based biological function, cellular components. Those are associated with the genome using that Swiss prot, PFAM, or signal IP and carbohydrate associated enzymes database. Then pathogenicity related gene, we can identify virulence related factors, their effector molecules. Then resistance to drug, that includes fungicide resistance also. And if we are talking about human pathogen, then you can analyze the drug resistance to particular drug, and then antibiotic resistance in case of human pathogens. Then comparative analysis, we can compare the Centeny. Using Centeny, we can analyze the evolution of the pathogen and divergence time. Then gene family. Then we can have phylogenetics of that particular genome across the family or available genome associated with the closely related to that species or genus. Next, please, sir. So, how the gene we can analyze a particular chromosome or particular gene of that particular genome of the pathogen. One example is Haemocysteria who has core chromosome also and accessory chromosome also. And these, there, if we talk about chromosome number 7 of this Zymoceptor which is right arm is highly enriched with chromatin associated histone, and is poorly transcribed. But gene cluster that has potentially evolved in uh, production of secondary metabolites and transcribed at the right arm of chromosome is majorly to the accessory chromosome, and they are highly divergent. Next, please, sir. Then how to analyze the hybridization between or emergence of a new species due to hybridization. When genome of two species of Plumeria graminis were compared, uh, and to know the emergence of Plumeria graminis triticale. So triticale is man-made cereal, but earlier it was not susceptible to Plumeria graminis. But due to hybridization between Plumeria graminis tritici and Plumeria graminis caecalis, um, this initial hybridization and backcross that happened in uh, nature itself has emerged of new species that has become virulent on the man-made cereal called triticale, and species was named as Plumeria graminis triticale. Next, please, sir. So, horizontal transfer of virulent gene is the one of the most discussed example, and tox is the most discussed gene that has identified as horizontally transferred between uh, three species, that is Pyrenophora graminis tritici, then Staganophora, Parastaganospora nodorum, and then to Bipolaris sorokiniana. And this event was earlier horizontal transfer from Pyricularia graminis tritici to Parastaganospora nodorum was recorded in 2006, and after around 15 years, the another genus jump has been identified in 2020 and 2018, first in Australia. The gene tox-A was found to be virulent, or found to be a virulent factor in Bipolaris sorokina, first in 2018 in Australia, then 2019 in USA, and in 2020 in India. So this is the comparison of genome of particular three pathogens. Those includes the Pyrenophora tritici repentis, then Parastaganospora nodorum, and Bipolaris sorokiniana. And this particular segment of 130 amino acid, we include plus some transposable element on both ends, has been found to be jumping or horizontally transferred between these three species. So the region is includes 12 kb homologous region that is present in all these three species, and shows differences in sir, next please sir. So in total alignment of this region includes 12 kb homologous region between these three species, and this shows differences in AT-reach region, that is given in green line, and large increase are denoted by a small red asterisks in the genome. The decay the decay in identity near the edges of region can be attributed to the repeated induced polymorphism, and all annotated genomes are shown using the accession number of Parastaganospora nodorum in green bar. So difference in promoter region as a result of presence of small indel, and there is 138 base pair insertion, and has no homology with Parastaganospora nodorum. But in Parastaganospora nodorum has only 43 base pairs out of 234 base pairs are present. And if we compare this Parastaganospora nodorum with Bipolaris sorokiniana, it has only 148 base pair indel is present, which is nearly comparable. Sir, next please. Next slide. So we can see here how this small portion of tox A is being transposable from has a difference between Parastaganospora nodorum, Pyrenophora tritici repentis, and Bipolaris sorokiniana. This particular region is present on chromosome 8 of Pyrenophora tritici repentis, but Pyrenophora nodorum and Bipolaris sorokiniana chromosomes are not identified, but that particular region is being transferred from one genus to another genus. Next, please, sir. So we can see the comparison between these three pathogen and outlier as Parastegonophora aviaria tritisae. So this is how the divergent isolates of tox A are being evolved together. So next, please, sir. So using this information, we further try to get insights about the different haplotypes of tox A gene across the fungal species. So till now, more than 24 24 haplotypes for the tox A gene across the three species have been identified, and they were normally present in several various isolates of the particular species. So, if we talk about India, in India, tox A1 haplotype is more prevalent. And if we talk about Middle East, there are most of these tox A haplotypes are prevalent. Next, please, sir. Then there is another case study of global genomic analysis of powdery mildew fungus that is associated with pathogen spread. And this case study shows how this fungus migrated from fertile crescent during the wheat domestication to Eurasia, then followed by America, and where it hybridized with unknown grasses, hybridized in unknown grasses, powdery mildew species, and again evolved, and then trade between USA and Japan and other Asian countries bring the USA strain to the Japan and Eurasian strain to the China. We can see in next slide how the species were divergent and how this trade was trade brought the species from one place to another place. Next, please, sir. So these are the diversity among the species and the comparison of species. These study includes comparison of 172 genome sequences of uh, Plumeria graminis tritisae strains across the globe, and those belongs to Eurasia, then Israel, then USA and Australia, then Japan, and China. So you can see the diversity among the genomes within the species, and those are evolved differently just because of their hybridization with Plumeria graminis species with some species on the grasses, and likewise. So each country has made different clade when compared more than 172 genomes. Next, please, sir. You can see how these species transferred the uh, uh, Australia, and the Eurasian species will move to China and some Middle East countries. Next, please, sir. So this is the one most popular project currently ongoing is the thousand fungal genome project, and coordinated by this fungal genome resources consortium, ZGI, and Myco. This website particularly has more than 1,000 genomes available across the various fungal species. Next, sir. So this is the website of Thousand Genome Project, and this has various resources available here. Includes the outcome of this Thousand Genome Project, and resources for comparison of genomes. Those are available within this project. So we can go to this website and directly compare the genome of our interest with our sequences also. Next week, sir. So this 1,000 genome panel has retraced the global spread of the pathogen and adoption among the major fungal crop species, and included most studied genomes in this project included Zymoceptoria tritici and Plumeria graminis, followed by Puccinia graminis tritici, and then a few other fungal species. Next, sir. So apart from these serial pathogens, we are engaged in genome sequencing and reporting the first genome of some species associated with vegetable crops. Includes Cercospora brassicicola, Cercospora citrulina, Cercospora sesame, and Pseudocercospora cruenta. Currently, we are sequencing several strains of Bipolaris sorokiniana to know the divergence and evolution of this Bipolaris sorokiniana within the India. Next, sir. So our genomes have been the Cercospora genomes are now available as reference genome. Those include Cercospora citrulina, Cercospora sesame, and Cercospora brassicicola, just because they were first genome and now are publicly available. Next, please, sir. These are the assembly link and accession numbers of the genomes. Next, sir. So this is the comparison of genome. This is one example how we can compare the various genome to know the differences or divergence or evolution of new species, if any. So if we talk about Cercospora genomes, they are within the genus, the diversity is very less. They are highly complex and uh, they're they are not so much diverse compared to any other. Next, sir. So we try to compare the distribution of orthologs and protein data within genome. So if we see the comparison between one serial fungus, Cercospora zmidis, and uh, those are associated with uh, other pathogen, the that is most closely associated or has least or not much diverse from Cercospora beticola, that is mostly infecting several vegetable crop and has a diverse host range. Next, sir. So we can see here we compared number of unique proteins among the Cercospora species, and most of them, around 8,000 proteins are found to be similar across the species, and only 112 proteins are unique among the species. Next, sir. This is where we can publish, or where we can find the published genome of the plant pathogenic fungi. Includes four most popular journal, that includes Phytofrontiers from American Phytopathological Society, has APC. Uh, Only a journal called IMA Fungus does not charge any processing charges, but 4 to 5 genomes are together clubbed and then published and available in a public domain as free of charge. Next, sir. So there are some GitHub resources. Those are available for biological visualization of the genome, and visualization of the tree. Next, sir. So these are some more databases, links to databases available for sequence analysis, protein and nucleic acid analysis, then secretome, then signal sequences, signal peptide cleavage analysis, likewise. Next, sir. Next, sir. So these are some more links apart from NCBI and ZGI website, specific for Neurospora, then Aspergillus nudulans and Aspergillus flavus, Magna porphyracea, and likewise. Next, sir. So this is one interesting paper I just put at the end. This includes the genome scale phylogeny of fungi. This has been published 2 years back, but this compares more than 1,644 species, and this is based on the 290 genes only. To know the divergence and phylogeny of fungi across the several fungal species and classes. So this is all about today. Next, sir. Thank you. We have several partners. Those help us in funding and encourages us for this kind of work. Uh, so thank you. Hello. Hello.
Yeah. Yeah. Thank you, Dr. Sudhir. Thank you, Dr. Sudhir, for a very nice and informative talk. I think before we go to the questions, I have a couple of questions from my side. That have you
Yes.
tried to correlate the genome-based taxonomy on Cercospora with the present kind of polyphasic taxonomy that we follow? So, does they correlate? Have you tried to work on that or evaluate on that?
Actually, before sequencing, we did polyphasic taxonomy to confirm that particular species using 2 to 3 barcodes, except ITS plus 3 markers. Like colmedulin or GAPDH plus SS SSU and LSU.
And
So
okay, fine. And were there was correlation, means the taxonomy that you obtained by the genome sequence was correlating with the
yes, yes,
that
yes,
you were okay, okay. So
yes, yes,
another
yes.
question for the benefit of the audience, I want to ask is that the kind of resources that you have mentioned, so they are sufficient for if one wants to go and have a kind of do the genome analysis. So those resources that you mentioned by GitHub resources, that they are sufficient. Once the pipelines are already there, you can just go there and work based on the instructions there and analyze the data.
Yeah, but actually the thing is there you need to do some modification based on your sequence information or sequences you get. What kind of sequences are there? Because when we do the sequencing, if you go for paired end data, or if we go hybrid assembly, then sequences we get are different. So based on that, we have to have small modification. But most of the GitHub resources are they need not to have major changes.
Okay.
You have to have changes like file upload and type of file they are asking.
Okay, okay. And is there any kind of workshops available within India for those who wants to work on fungi can attend and then get to know about that thing?
Yeah, actually, IIT Hyderabad and had a workshop on this genome analysis and like that. So there
Specific,
are
specific to
small
fungi?
workshop.
Sorry,
No, they
specific to
were
fungi?
that was for genome comparison and genome analysis only, not specifically fungi.
Okay,
So we can learn and we can
okay,
try it again.
okay, okay. Thank you. From my side, I think Dr. Shishupala is sir. Please go ahead with the question. Uh, I think Dr. Shnoy, you had a question from your side. Dr.
Sir,
Shishupala's
yes, sir.
mic.
Yes, sir. Yeah. Yes, sir. Sir, namaste. It was
Namaste,
a
sir.
wonderful,
Yes, you are audible.
yeah, it was a wonderful information. I just wanted to know, because plant pathogenic fungi, isolates
Yes,
and species vary in their virulence. When you
yes,
do
sir.
whole genome mapping, you will get the
Uh-huh.
sequence, but you will not
Yes,
know whether the particular gene responsible for toxin or pathogenesis is expressed or not.
yes. We need to have that further analysis when we go for, actually that's a long process, because in Sarcospora what we are doing is we identified most identical proteins. Those were like 112. Again you have to analyze one by one, so it's like it's long
Okay.
thing to do.
Okay.
But
With respect to,
if we know go for genome wise or genus wise, Cercospora, there is known pathogenic factor cercosporin, but we know that the gene related to cercosporin. Then we have to identify something not related to cercosporin, but the protein that is infectious. But again, that needs one gene, one genus needs a long process, because if you identify, say for example, 20 proteins, those are predicted as a effector molecule. Then you have to
okay.
test every protein.
Okay. Sir, one more
uh,
thing.
yes.
The protein of the pathogen recognizes the
uh,
protein of the host in terms of host specificity. One of
yes.
the molecular interaction between fungus and the plant. So in
Yes.
such
Yes.
case, most of the plant genomes, particularly crop genomes, have already been worked out. So
Yes.
will it be possible? Will it be possible for identifying host recognition factor with the
Yes,
genome
it is
mapping of?
possible.
Okay,
Yes, it
okay,
is
okay.
possible. But that is majorly associated with biotropic pathogen only.
Okay,
Necrotrophic
okay.
pathogen,
Yeah, yeah.
necrotrophic pathogens are not host specific.
Yes, sir. Yes, sir. I
So
understand.
or even hemibiotropic pathogen like Zymoseptoria has
Downy
some host
mildews,
specific.
downy
Yes,
mildews
powdery
and rust.
mildews, rust and powdery mildews.
Yeah, yeah. Sir, in case of
Yes,
bipolar sorokiniana, you had
yes.
showed in the slide mixed isolate. What do you mean by that?
Mixed isolate means we have several isolates. Those vary in terms of like colony morphology.
Okay.
If we go for colony color, some are black isolate and some are mixed, means colonies are not dark brown or black. So
Right.
we used mixed isolate because they are not different based on, or they are not different species. They are the same species, but
Because
the changes are just only in melanin content of that particular colony. That's why that
correct, correct.
so we
Because
thought
most
it
of these pathogens are asexually reproducing.
yes, yes, sexually reproducing only, because
Yeah,
in
not much genetic
bipolar,
recombination possible except for the mutation.
yes, in bipolar serociniana, sexual phase is rarely known and rarely reported,
Yes,
even though it's hemibiotropic pathogen.
yes, yes, yes. Thank you, sir. Thank you.
Yes,
Yeah, yeah. Dr. Shinag,
Yeah, thank you, Rohit. Can you hear me, Dr. Sudhir?
yes, 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 sir.
That was a wonderful presentation. Thank you very much. We would like to have whole genome information in fungal taxonomy, and have a more predictive classification system for many groups of fungi, especially plant pathogen fungi, maybe human pathogens. But see, if
Yes,
you
yes,
consider the situation in India, people say we don't have money to buy a sequence IQS also, except for rich labs like based at Pune or Chandigarh or somewhere else.
yes,
Okay,
sir.
so
Yes, sir.
but of course, having sequence 8 genes for culture trichome, but I understand there is a potential in improving the taxonomy when we go for whole genome information. I mean, how do
Yes,
you
sir. Yes.
foresee the future where we can incorporate the whole information along with other omics data, and also consider the cost of experiments and cost of quality manpower. I mean, it's a very long question. I would like to get your
Yes.
response on this.
Sir, actually, I see if you talk about this genome sequencing and all. First, in case of plant pathogens, I see we need to have these pure cultures in several repositories. That is long missing, because if you go towards the repositories, there are very few cultures available of plant pathogens, and that I expect we can have this sequencing and data repositories through these large repositories like in Chandigarh or in Pune, because they are specifically meant for this conservation and making data available for public domains and all. That is my the picture that comes in my mind when I look as a future of genome sequencing. Because if you go for small genome, it costs nearly about 1 lakh. That gives only information, not the bioinformatics analysis or insight to the genome. So it cost is very huge.
Yeah, that's why I asked. I mean, as the time progresses, the cost might come down. So,
Yes.
what is your a rough guesstimate, or when we could be easily incorporating this whole genome information in fungal taxonomy in next 5 years, 10 years, 20 years? At the global level, the trend is already there. But India has to pick up. I mean, India is still struggling with the one gene, one multi-gene. So, what are your final remarks on this for me, please?
I expect next 5 years is the will change everything, because when I started working with this in 2016-17, cost was more than 1.5 lakh or around 2 lakhs. Now it has come down to 90,000 to 1 lakh rupees. So I think 5 more years will help us to get through this, because in if we talk about Europe and Australian countries, they are sequencing. I take one example of Zymoceptoria. They are sequencing more than 200, 300 isolates at a time, and they are doing analysis of genome-wide association studies and likewise. So we are lagging far behind that. But I think next 5 years, maybe 2030 or before that, we will have the drastic change into that. That's my final.
I thank you very much for those insightful comments and response. I'm sure you'll be torch bearers for fungal genomics in future. Thank you.
Thank you. Thanks.
Thank you, Dr. Shna. I just like to add a little bit. I think the culture collection of, like in prokaryotes, it is happening done by DSMZ, and many of the culture collections, like culture collections, can have the fund from the funding agencies, of course, maybe joint DBT, DST, or CSI joint kind of fund kind of thing, and whatever new species we are publishing, so those as a kind of means complementary, the culture collection should take the initiative to sequence the genome on as compared to that will be my opinion kind of thing. And this kind of things activity are being
Yes,
done for
yes.
the prokaryote and bacteria like that. The moment you deposit
Yes,
the
yes.
culture to a culture collection as a new species, then on as a complementary they do genome sequence for you like that. So something like that should
Yes,
be done in the Indian scenario also, to have
yes,
this thing,
yes,
because as Dr. Shnoy rightly mentioned, fund is a problem for average lab in India. Yeah. Thank you. Thank you so much,
yes,
Dr.
yes,
Sudhir.
yes, yes,
Uh,
yes.
yes, Dr. Poonam, please go ahead with your question.
Thank you, sir. Thank you, Sudhir sir, for your this presentation. Very informative session it was. Sir, my question is pertaining to whether the plant which are being having the resistance against certain pathogens, or basically I am talking about the elicitor protein, can they have the induced resistance in the plants through the epigenetic modification, or epigenetic kind of mechanism is also involved there?
Yes, it's there. But
And it,
major resistance in plant is because of its genetic structure, and epigenetics is something comes with genetic imprints through evolution of that particular species, or how that plant is exposed to the environment and other thing. But the majority of the resistance is due to genetic structure. When I say genetic structure, that means number of resistance gene present in that plant. If you take example of it, there are more than 70 resistance genes for leaf rust, more than 50 resistance genes for stem rust, and around 70 resistance genes for yellow rust. That's how it works against the pathogen. But majorly, this resistance imparted in the host is against these biotrophic and hemibiotraphic pathogens.
okay, sir. And one more question. And if we want to detect, if we want to do the genetic analysis, that what could be there for such kind of resistance, then what would be the easiest way to do those experiments? How we can do for that?
That particular plant, inoculate your target pathogen
At the physiological levels, we have the insights, but
pathogen
we,
of the plant and test its resistance against that
okay, sir,
pathogen.
at the what I say, at the morphological level, we have the insights that there is a resistance, but if we want to check for the genetic pathway, what would be the way by which we can determine the pathway through which that resistance is being incorporated in that plant?
Then there are other methodologies that includes those are very costly methodologies, but includes like transcriptome analysis of the
Okay,
plant or epigenetic sequencing of the plant.
okay,
But those are very long process and includes very huge cost. But simply we can do is run some gene specific markers
okay,
in SSR, and you can identify which gene is present.
okay, sir. Okay, thank you, sir. Thank you so much.
Uh, uh, yes, Vritti Naik.
Yeah, hello, sir. Thank you for the informative talk. I have a very technical question actually. How is the
Uh-huh.
genetic genome comparison actually done using which platform? Can you elaborate on that?
Genomic comparison has several means several methodologies actually. But when I used to first have this phylogenetic analysis, then to compare genome, and then comparing. I use R software and several tools into that. Actually, verbally, it is quite difficult to tell you how we compare one genome with another genome, because
Yes, yes.
that in
Is there a particular pipeline that you used or a
yeah,
different
yeah, I can
combinations
actually
of
share.
different softwares? Okay, okay. Yeah, that would be nice, sir. Thank you.
Yeah, I can share
Yeah,
you that.
yeah, also another question which you have already answered, but is genomic comparison it alone enough for correlating all the characteristics, or expression studies are also necessary for this?
Actually, you can predict using the genome analysis which proteins are unique, which proteins are uh, their nature is of related to pathogenicity or effector molecules. Then you have to prove that effector molecule using implanter techniques, because that is not
Exactly.
sufficient to tell. If, say for example, toxin X you identify as a effector molecule for say for example bipolaris urociniana, but you need to prove that that toxin X is required to cause the disease in particular host. That for that you need to sequence that protein, then artificially you need to secrete that protein in some host like Pichia pastoris or something like that, and then that protein molecule, individual protein molecule, you have to infiltrate into the plant to see whether that protein is necessary for disease or not. It is causing
Exactly.
symptom or not.
Yeah, that was my question actually, because most of the times predictive functions are not enough. You have to prove that it is happening. So
Yes,
that was
yes,
the
yes,
query. Yeah.
yes.
Okay, thank you very much, sir. Thank you.
Yeah, I think Mr. Naik has a valid question. So we'll in sometime we'll try to have Dr. Sudhir or some or more persons to have a kind of a mini workshop kind of thing for the analysis of the genome. Thank you so much.
Yes, yes, yes.
But sir, please go ahead. Please go ahead with your question, sir.
Hello. Yeah, thanks, Rohit.
Yes.
No, I just wanted
Namaste.
to say thanks very much for this, workshop, this lecture, because Dr. Sudhir spoke so well, and every step is actually a learning for me. So I just wanted to give my appreciations for his talk. Wonderful it is. Thank you very much.
Thank you very much, sir, for your word. That is so encouraging for us.
Thank
No,
you.
no,
Thank you very much.
it's truly nice, Sudhir, to hear this, because we really need more such exercises in India, because as you said, suppose a culture is deposited in the repository, if the basic sequence is known, I think it will be a good
Yes,
reference material for others also. See,
yes,
instead
yes,
of people individually taking this exercise should be done by the repository, like IMTech, the ITGC, or even in Agarkar Institute. But we, as you said, probably it will take another 5 years or in the coming days. We look forward to that, and then more
yes,
people will participate in this exercise. I think that's
yes,
my great wish. Yeah. Thanks very much.
yes.
I'm very happy with
Thank you
you.
very much.
Presentation. Yeah.
Thank you
Thank
so
you
much
for
for
your
the
words,
kind
sir.
words, sir. Thank you so much. Yes, Revati, please go ahead with your question. Sorry. Yeah.
Hello, sir.
Yes,
Good
ma'am.
afternoon to everybody. So I'm Revati. I have a question is that to Sudhir, sir. So
Yes.
how to identify or confirm if a newly seen fungal species is pathogenic or non-pathogenic? Only genomics or molecular sequencing are what are all considered as the gold standards. So could you please elaborate, sir?
Yeah, actually, whether it is pathogenic or not, that you cannot identify just using the genome sequencing. You need to prove its pathogenicity using a technique called Koch's postulate. You have to inoculate that fungus on the host from where you isolated. You have to confirm it is causing symptom on that host, and then again you have to re-isolate from that host. So that needs implanta experiment to confirm it's virulent or pathogenic or not pathogenic.
Can I come in, Dr. Rohit?
Yes, please go ahead.
Dr. Rohit, this is a very interesting question because the cost postulate might take 2 to 3 days at least, right?
Yeah,
Yeah.
2 days.
Yeah, but that probably we could, I mean, we wish to have a very rapid technique, maybe which can be done in 1 hour, which can tell this what is the possible particular fungus being pathogen at that particular moment, Do you foresee
Yes,
such,
Uh-huh.
yes.
do you see foresee,
Time effective it is. Yes.
yeah, do you foresee such a future, Dr. Sudhir, with not only with genomics, all the kinds of omics tools actually?
Yeah.
Hello, sir. I have one more thing to ask you. So you said that to do a whole genome sequence, it will cost around rupees 1 lakh. So could you just elaborate? So where it is done, what are the labs, and what do you think that we should do to bring the cost down?
Bring the cost down. I don't know, but because the kits involved in that sequencing technology, that preparation of library and sequencer cost and cost including manpower is taking us towards 1 lakh actually. Because most of the kits are imported, and then there are some companies has monopoly on those kits. That's why the costs are going high. And the sequencing is done in several institutes, like in Pune, there is NCCS is doing whole genome sequencing. Then this Chandigarh lab is doing whole genome sequencing. Then there are several private companies also, they are doing whole genome sequencing, like Genotypic Technology, AgriGenome, and likewise. So you
So
can approach to anyone.
So just one more thing. So you said that to add in, so as if when you see a new fungal species, it is not easy to identify whether it's a pathogen or non-pathogen just by seeing it. But you said that not by doing whole genome sequencing. You said the method. Could you just spell the word, please?
Koch postulate. Not easy means it's easy. Proving Koch postulate is not so tough. It takes 2 days. That's it.
Okay, sir.
But
Thank you, sir.
you
Thank you
need
so
to
much.
have the host ready for your test. That's it. It's not so tough. Means
I'll just add a few points to it, Ms. Revati, because what Dr. Sudhir has said is true. But if you know the analysis, then the sequencing you can do around 7 to 8, 10,000 per GB. So around, if it is 4 to 5 GB, it will cost around 50,000 rupees for sequencing through nanopore, and then you can analyze by yourself. So in this way, you can reduce the cost. So that, and if you analysis you are able to do, then the cost of analysis will be reduced. So that's why I think it will be good if you can have attend a workshop and try to learn the analysis, genome analysis kind of thing. Yeah. So thank you, Dr. Sudhir. I think, Dr. Shino, most of the questions mentioned on the chat box are covered through the verbal questions. I think most of the questions that were mentioned.
I think Dr. Rohit, it's not a bad idea to keep some questions unanswered, maybe for the future. I think we can discuss some of these questions in our platforms, MicroIndia or MicroAsia platforms, if people are around. There will be more people getting benefited and giving input out. I think.
Five people.
I will write down.
Sure. So you want to have any final comments from your side about it? And you want to brief about tomorrow's seminar also, or conference?
No, I think, when I was a PhD student, people were already expert in morphology. We were young people. We would learn the things fast and implement. Then the already morphology experts, they were finding it very difficult to transit. Shift from morphology and embrace molecular data. So my situation is that I am finding it very difficult to embrace the newer technologies like whole genome information, the artificial intelligence. I think it is good to have an open mind about the new techniques. If you cannot learn yourself, have your team member learn it and implement in your lab. I think best example is my PhD advisor, Kevin Hyde. He embraced all these technologies. He's a great morpho expert, but he embraced all kinds of technologies and got them implemented in his laboratories. And he's one of the legends, living legends of mycology. So we should be open to new ideas. And if those ideas may be uncomfortable, but these ideas may take you to the truth, the truth what you have just so far understood. Actually, that's my comment. Thank you so much, and Dr. Sudhir, wonderful talk. Good to have you again, and we will try to have you one more time, maybe two more times in this year or next year, or some technical aspects of it, I guess. Thank you so much, Dr. Sudhir.
Thank you so much for your words, sir.
Small things I'll add, like Dr. Bhatt and Dr. Sridhar has also been remarkable, sir, because they have been listening to the advancement techniques. It's so interesting and trying to learn at this stage also. So we always get inspired with that. So youngsters should also do not leave the morphology, phylogenetic thing, and now comes the genome analysis. Yeah, thank you, Dr. Sudhir. Thank you very much for your time and the things. Thank you so much. I think feedback link will be shared with all. So kindly fill the feedback in the next. Dr. Shna will be sharing it on the chat box. So there you can just fill up the feedback form, and based on that, we'll be giving the certificates from CVAC.
Rohit, I just would like to add one more thing. Actually, one of the reasons we get enthusiastic about conducting these kind of programs is because of the audience. We have a set of wonderful audience. For every online talk, we get around 100 people. That on Saturdays and Sundays. I want to specifically thank our audience from MicroAsia, MicroIndia. Association of Fungal Biologists, and maybe from followers of CPAC, or people who are not at all associated to any of these platforms, but maybe got to know about our programs. And I want to thank them. I thank our audience for your wonderful participation, and be supporting us whatever we are doing. And your encouragement is is our actually strength. Thank you so much.
True. Thank you. Thank you, Dr. Shna. Thank you for the kind words. Yes, I completely agree with you. Yeah.
Thank you. I think with that we will just say sign off for the day, and we'll meet tomorrow for the international seminar on fungal biotechnology and informatics. Yeah. Thank you. That will be at
Bye.
Yes.
10 a.m. Indian Standard Time.
Yeah. Good for the day. Good for the day.
Thank you.
Okay. Thank you. Thank you, sir.